My name is Bart van der Vossenberg and this is presentation 1.6, Assembly of Sanger sequence data of the Practibar course package. During the Practibar course, background information and hands-on training is provided for the use of DNA barcoding in the framework of plant health. In this presentation, we are going to look at the assembly of electropherograms. First, we'll address the question, why do we need electropherograms or trace data? And then we'll look at generic de novo assembly workflows. With these workflows, we are going to look at a couple of examples in Genius. If you would like to learn more about electropherograms and how they are generated, please look at presentation 1.4, DNA Barcoding Wet Lab. Now, why do we need trace data? When you have Sanger sequence data generated, the data becomes available in a number of file formats. For instance, you have the SCF format, the ABI or AB1 format, and the FASTA format. So the first two file formats, the SCF, ABI and AB1 files, they contain trace data or electropherograms. The latter file format, the FASTA format, is just a flat text file format and does not contain any information on the quality of your sequence run. We can demonstrate that with an example. This is a stretch of sequence generated from sample 1 using the forward primer. You can see the nucleotide sequence A, G, G, T, G, C and so on, but you cannot tell anything about the data quality. Now, if we would look at the electropherogram that produced this stretch of sequence data, you can actually see that this is very, very poor data, and you would like to reconsider including this data in your analysis. When you're assembling trace data to generate a consensus sequence, typically you have two reads, one from a forward reaction and one from a reverse reaction. And when the sequencing primers were the same as the ones you've been using for the PCR amplification, then you know that one of them is reverse complementary orientated of the other. But what you get from your sequencer is two sequences in 5' to 3' orientation. So both from your forward and your reverse primer, you have sequences in 5' to 3' direction. And of course, since the DNA is reverse complementary orientated, we have to reverse complement one of those two reads, like so. Then you can assemble them, and the overlapping regions are used to generate a consensus sequence. And actually you have confirmation of a given nucleotide from one run in the sequence of the other run. And also, when you have poor data, and actually the blue graph that you see in the background, that is the Phred score. So if you have low Phred scores here, you have sequence data with a fairly high Phred scores here that could help you resolve what is going on in this section here. So the overlapping sequences are used to generate a consensus sequence. But still, it is important to visually check the assembly and the consensus sequence generated. For instance, if we have two bits of sequence data in FASTA format, we can see that they are different. So 4 A's, 2 T's, a G and an A from the forward reaction, 4 A's, 1 T, a G and A from the reverse reaction. So if we would align this, we really wouldn't be able to tell if there would be 2 T's at a certain position or just one. We need the electropherogram data for that. So this is the trace data from the forward reaction, and this is the trace data from the reverse reaction. So when you look at the forward reaction, you see nicely separated peaks, no or very little background signal. The 2 T's are very clearly visible here, nicely separated. And when you look at the reverse reaction, here something has gone wrong with the base calling. We have 4 distinct A's. The 2 T's are present, but it is poorly separated from this G signal, resulting in only 1 T in this sequence read. So if we would align that, then actually we could see, okay, these 4 A's align, one of the T's align as well, but actually looking at these double T's here and these double T's there and this G and A, we can come to a consensus sequence of 4 A's, the 2 T's, and G and A. In the APO standard, we've given some guidance on how to visually inspect your assembly and to draw conclusions on what is a reliable consensus sequence. So additional trimming that might apply, using ambiguity codes for double peaks, how to work with single insertions or deletions in coding sequences. I'm not going to read it out loud for you here. You can either pause the video or look at the APO standard, and I think the latter is best. Should there have been some updates, but these recommendations will be discussed directly or indirectly when we'll look at some of the examples in Genius. So this is the generic assembly workflow. First you upload your electropherograms in the analysis software. And here you can see electropherograms of a forward and a reverse reaction. In peaks, the blue peaks again is the Fred scores of your sequence data, and typically in the beginning of your sequence run, the quality is a bit poorer, and then you have high quality, which if your sequence run is long enough will slowly fade out. These two you assemble de novo. Then you trim amplification primers and untemplated DATPs at the end. If needed, you can perform some additional trimming if the quality is low. Then you manually inspect all the conflicts that are there in the assembly, and then from the assembly you create the consensus sequence. In Genius we have 2 examples prepared to demonstrate the assembly of sequence data and the generation of a consensus sequence. So the first sample we have is a COX-1 amplicon from a beetle, and so the amplicon was made using primer pairs LCO 1490 and HCO 2198. People could recognize those as the standard CO1 barcoding primers, and for each of these samples we have a sequence read, which are shown below. So if I select, for instance, only this one, the LCO reaction, there you can see the sequence read, and I'll zoom in. So below here in the different colors blue you have the sequence the sequence itself, and then in the different colors you have the trace data or the electropherograms, and then in the bars in the back you can see the fret scores. So when the fret scores are high, you have nice light blue colors, you see very nice peaks, well separated. When the separation is slightly less and you have lower fret values, then you see a slightly darker shade of blue. And when you have sequence data of poor quality, then a dark shade of blue is used. And also you can see that in the fret scores. So those are just different ways of indicating the quality of a Sanger sequence read, and this will help you to assess the overall quality of the assembly and the consensus sequence that you're going to generate. So both sequence runs are represented in a 5' to 3' direction, and typically in the beginning the quality of the sequence is poorer, and then you get good sequence data, and depending on the length of your read, this will tail off to slightly less quality of reads. But from a distance this looks very nice. So in Genius I can assemble this by clicking on the Align Assemble button, select De Novo Assemble. I use standard settings for the highest sensitivity. I do have some options here for trimming primer sequences. And actually I'm trimming here, I let the software search for the primer sequences, LCO and HCO, and let them automatically trim them or annotate them as trimmed, so that they don't appear in the consensus sequence. Okay, save the contigs, press OK. That was very fast. And here we have another file now, which is the assembly of these two reads. What you can see, there is a highlighting option in Genius. So if I turn it off, basically you see all the bases in the assembly. But when I turn it on, it highlights all bases that of all disagreements to the consensus sequence. So there's a consensus prepared on top here. So there's not too much conflicts. There are some gaps here we have to look into. But also what is immediately noticed is that we have these annotations here now in green. So actually that's the primer annotation of the HCO primer, and you can see the purple of the pink annotation is a trimmed annotation. So this section here also is excluded from the consensus sequence. And on the other end we have the LCO primer, also primer annotated and trimmed, so it doesn't participate in the consensus sequence. Okay, so one of the things we have to check is the translation. The translation is really important, an important aid, because you could maybe ask yourself, are these deletions, are these true deletions, or are this A and these two A's here, are they really there? Turning on the translation of the sequence helps you in assessing if something went wrong. So I'll just turn on the translation on the consensus sequence. We are working with the invertebrate mitochondrial code, and the COX-1 gene has a reading frame, a starting codon in reading frame 2. So what immediately stands out, and I'll zoom in a bit here, is that we have a stop codon. So and that's actually not because we have the wrong code or the wrong frame, but actually the direction of this fragment of this assembly is reversed. You see the LCO reaction, and in this LCO reaction you find the HCO primer sequence, and here you had the HCO reaction with the primer physically being here and initiating the Sanger sequence data from that point. But actually the HCO primer is a reverse primer. So we have to, and I'll switch this off, we'll have to reverse complement the entire sequence, the entire alignment. And I'll save that. If I now again turn on the translation, invertebrate mitochondrial, frame 2, then we have a nice translation of this sequence without any internal stop codons. So internal stop codons would indicate that you have maybe a pseudogene, or that you have artifacts in your assembly or in your sequence. So this we have to resolve. Nice, the quality, yeah. So, and you have very good sequence data in the opposite run that will allow you to determine if this overall consensus sequence is reliable. And this actually looks really nice. Yeah, like so. So this looks really nice. And now also in the consensus sequence you see the different colors blue used, which is actually the base with the highest quality that is attributing to the consensus sequence here. So from this I'm going to generate a consensus sequence. I use the same settings as I had just now, and this results in a 658 basis consensus sequence. And just looking at the color, I can see that the overall quality is very high of this sequence, and this I could use in the follow-up in the analysis using online databases. So that was example 1, and now we'll turn to sample 2. So again, sample 2, we have two Sanger sequence reads. This time we have sequence data obtained from a plant sample, and the partial RBCL chloroplast gene was amplified using primers RBCL AF, which stands for forward, and the RBCL SI reverse. And what you can see from the sequence data, the quality of the sequence data when I select both of them, is that, and you have to remember that these again are in both in 5', 3' orientation. So here at the 5' end we had a primer initiating elongation, incorporation of the DDNTPs, which results in the Sanger sequence data. And actually, so in this reaction, in the RBCL-AF reaction, in the end of this primer sequence, we expect to find the primer RBCL-SI reverse. And the same goes for this reaction, initiated with the SI reverse primer, and in the end of this sequence, the 3' end, we would expect to find the RBCL-AF primer. So basically, this will give us some headaches, probably, and we already noticed that the quality here is rather poor, and we have to take special care in assessing the assembly and generating the consensus sequence. So the steps are the same. De novo assemble. And this time I'm going to trim the sequences with the primers SI reverse and A forward. Save contigs. Okay. There we have the assembly file. And notice that the highlighting button is on. So actually here in these darker shades of blue you see conflicts between the two reads, and the highest contributing base is selected in the consensus sequence. So basically these lower quality bases conflict with the sequence at those positions. So we have to look into them. Again, translation will help us to determine whether or not we have insertions or deletions that shouldn't be there, that can help us to improve the assembly. But first, let's take a look at the orientation. So we here have the reverse primer at the beginning, and at the end we have the forward primer. So also here we need to reverse complement the entire alignment. Okay, like that. And then we need to take care of the translation that is listed here. So it is set on standard. Just now we used the invertebrate mitochondrial code, but for this we use the bacterial code, because this is a chloroplast gene, and chloroplast genes use the bacterial code, which is translation table 11. And for this particular amplicon, it is frame 2 that results in the correct reading frame. So we'll just start with looking at the sequences here. So really this sequence data is really messy, and it doesn't help us a lot in terms of using this as support for the sequence data that we have in this read. So since the sequence data in this read is really nice, we can use only this single sequence to determine the consensus sequence, because this C and this T and the G and C and T and C over there, that's just messy data that wasn't called correct, so you cannot use it. But we can, for the stretch where the sequence data in the above read is really poor, we meticulously look at the electropherograms of the bottom read, and actually that looks really nice. Spacing is nice, nice peaks, equal height, good Fred scores, so this looks really nice. And actually now we came into a stretch where actually we do have good sequence data in both reads. We just continue reviewing this assembly, and then the quality drops in the lower read, and the quality in the upper read is still okay. So we're using now mainly the information from the upper read to look if that sequence data is okay. It looks really nice, nice peaks again. This deletion here or insertion, just the way you look at it, and the one here, I don't believe they are there, and actually the automatic consensus calling of this assembly also noticed that there should be nothing there. Yeah, so we don't have to trim additionally, because since we are using this setting of using the base with the highest quality to contribute to be more important in the consensus calling, then we are fine. So actually there is much more going on here in terms of poor data, but still this is actually fairly straightforward. We have to take a little bit of extra care
in the cases where the sequence data is poor in one of the reads. I'm going back and forth in the sequence also now to look at the translation. There are no internal stop codons. This sequence looks really nice, the consensus sequence. So I'm going to save this. I'm happy with the assembly. I'm going to generate the consensus sequence. Check the settings. Okay, so this is our consensus sequence, which looks nice. It's a 553 basis consensus sequence, which is actually what we expect for the RBCL locus. And again, this sequence would be used in the following steps for the analysis using online resources. And with that, we conclude the presentation on Sanger sequence data assembly and consensus sequence preparation. Thank you for your attention.